Okay, so we're going to be talking about PCR today, and one thing to understand is that PCR is basically a lab version of a process called replication, which happens in our cells all the time. So anytime you cut yourself and need to regrow skin, or you're just growing, literally getting larger, you're creating new cells. Every time you create a new cell, you're going to need new DNA for that cell, and we're going to make exact copies of DNA in order to do that. That's what we call in vivo replication. In vivo meaning in life, and then there's in vitro replication, which means in a test tube. So in vitro replication is going to be called the polymerase chain reaction, because basically we're going to make a polymer and then we're going to do that over and over and over again. So let me just start you off really quickly. So we're going to have a piece of DNA, and it is going to be pretty long. Generally, it's going to be in plasmid form, so in a circle, or it's going to be genomic DNA, which means it'll be, our 6-foot-long piece. We're going to heat it up, and that is going to destroy the hydrogen binds in between, pull that molecule apart so that it generates this shape right here. We're going to add in a primer. That primer will line up to this DNA and be complementary. So, for example, if this piece of DNA had the letters TACCA, this primer would then be ATGGT, and then the opposite would be true. So if this strand would be ATGGT, and this primer would be TACCA. Cool. Now you've noticed I put some arrows on this thing. So if you look at DNA, you'll notice that it has ends. So here's a double-stranded piece of DNA. And if we look at a single nucleotide, of which this whole thing is made, we're going to see that there are different parts. There is a phosphate group side right here, and that is off of a carbon ring here. And if we look at that carbon, orient it so that the oxygen is on top, we can count the carbons. So this is the oxygen, 1, 2, 3, 4, and 5. So if we look at this DNA molecule, we'll notice that these two strands either have a phosphate group on top, which is the case here, a phosphate group on top, or it will have a 3' carbon on top. So the phosphate group on top side we call the 5' side, and this side we call the 3' side. So you can see there's like a magnet right there that is going to, that symbolizes our 3' side and 3' hydroxyl. So our enzyme, in this case our Q5 enzyme, which is a type of enzyme from an organism called Thermus aquaticus. Let me start that over. So we're going to use an enzyme called a DNA polymerase. There are different types of that enzyme. The one we're using is called Q5. It is able to read long stretches of DNA without falling off, and it's pretty accurate. So different enzymes have different accuracy. So sometimes they will make a mistake and add in a letter that they're not supposed to add in. So the basics are, once this primer has laid down, because we've dropped the temperature just a little bit, the DNA polymerase will then add on to the 3' end one letter, and then another, 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 and so on, until it builds to the end of the strand, or in vitro till about 1,000 base pairs, unless we're using what we call a long-run enzyme. Okay, so this is the situation we're using for our circular DNA, because in our circular DNA, what we're doing is basically we're going to heat it, and when we heat it, the two circles are going to come apart. Our DNA strand, our DNA primer is going to anneal there and there. Right, so we have, let's say, TACCA there and ATGGT there. All right, so that polymerase then is going to extend and you'll notice that it'll just come right back up and hit the same primer in the butt. It's not going to be able to connect the two. It's going to actually leave a little bit of a nick. See that nick there and nick there? We can then heat these two things, and ideally they would come together in either the original plasmid or, oh, sorry, wrong color. And if you'll notice, we've offset the nicks just a little bit, so one would have a nick there, and the other would have a nick there, and that would allow this thing to circularize. Okay, so a couple things that we're going to do differently. First of all, our primer is not going to be an exact copy. It's going to be slightly different. So let's say that this is going to be a T. Let's make it green. And that would make this an A. So in this copy, we would have a T here and an A here. And here in our final copy, we'd have a T here and an A here. We'd have a C here and a G here. So this is the new copy. Ideally, our GFP gene is going to be, probably around here somewhere. So we've made a single nucleotide change to that gene. Basically, in our copies are going to integrate the primer into their sequence. So the primer is going to be part of the new copy. And we're going to add lots and lots and lots and lots of these primers, way more than we're going to have plasmid. So eventually, we'll start copying this and this. So this primer will sit on this primer will sit on this primer will sit on, basically, every copy that we generate is going to have our new nucleotide. But that doesn't solve all of our problems, because we're going to have some of this plasmid in here too. So after our PCR reaction, we're basically going to have all these versions. We're going to have the most of this by several magnitudes. But we're going to have the original plasmid, we're going to have two new copies, we're going to have one old copy, one new copy, and the other old copy and new copy. So the interesting thing to note is that all the DNA that I've kind of drawn in red or green is made in a test tube by a polymerase that we've added. And the black DNA is actually made by a bacterial cell. So remember, we got this plasmid by mini-prepping, which means that we took this plasmid from a bacteria, we put it into a bacteria, we let the bacteria replicate, and when it replicated, it made copies of the plasmid. And so all the black is made in a bacteria. And the interesting thing is that the bacteria is going to put little methyl groups on the backbone of that bacteria. We have an enzyme called DPN1, which will destroy selectively methylated DNA. So if we take this PCR reaction and we add a little bit of DPN1 to it, it should destroy all the old DNA. And get rid of this one completely, and leave us with our brand new plasmids. Our brand new plasmids have a single letter difference in the GFP gene, and hopefully that will turn our GFP, green fluorescent protein, into a blue fluorescent protein. But like I said, it's probably not going to work. So that's all the PCR stuff that you need to know. Cool.